So good afternoon to everyone. My name is Bryan Wang. I'm a postdoc, as Tom said, in the section of microbiology. And today I'll be presenting basically an introduction about 3D-omics that we've heard quite a lot about. And so this is a very ambitious title, but I want to remind everyone that it is a method development project, and so don't expect too much. In the audience, a lot of us are very used to doing traditional multi-omic things. And so the first step of multi-omics usually is homogenization. And so this is what we endearingly call poop scientists, which I also identify as one. But the 3D-omics pitch is really about how can we go about sampling in a way that preserves all of your spatial information such that you can still parse out host-host interactions and host-microbe interactions. And so this is done in a very spatially informed manner. And so this is the very ambitious workflow that we have come through. And you've heard a couple of these partners that are in this project. And I really like this project because it's a top-down approach all the way from animal sampling down to subsegmenting, cryosectioning, and cutting out really, really small parts of the chicken. And then also, if you were at the poster session, Nikolai, who's also part of the consortium, will take the data that we, as a consortium, develop and eventually do some modeling that's way out of my pay grade. And so in the first step of doing 3D-omics is actually we have to do the conventional macro-scale metagenomics. And so this is basically so that we know what is in the chicken. And so our goal is to go as small in a very spatial manner. And so the first step was to see who all is in a chicken gut. And so for this, we did traditional shotgun metagenomic sequencing, and we got up to 174 max. And so from there, we were able to now go into micro. Now that we know what's in the macro, we can go into smaller parts of the chicken. And so the workflow that my part of the project adopts is basically we sample after the animal trials, we take out the different organs of the chicken, then we snap freeze them, cryosection them, and then eventually we do microdissections using a laser, a very fancy microscope. And then the idea is to both do imaging, metagenomics, metabolomics, and also transcriptomics. And so this slide I had borrowed from Amalia in the audience because it's a very great workflow. Basically, after we get these microdissections from the chicken, we need to lyse and get all the bacteria and also carry out library building. And so the key to this is if we want to get to as much resolution as we can, we need to go and push our technology to as low of an input as possible. And so right now we're testing out library building techniques that can go down to picogram level of DNA. And so that is what we do. And from this, Amalia and Carlotta have generated a lot of microscale metagenomics. And then from this, what I hope you can appreciate is that both when you compare a cecum and the colon of a healthy chicken, the microbiota is pretty similar, which is kind of makes sense because there is flushing from the cecum into the colon. Okay, and so one other thing that they saw is if you compare different relative abundances of the different genuses, you start to see that there are co-abundance patterns that you only see at a microscale, which the macroscale metagenomics does not reveal. And so this was a very exciting evidence that we saw because it means that what we're doing is important. That like something that you do in macro you cannot see, that is suddenly revealed in the micro metagenomics. And so when I signed up for this conference, the scope actually talked about host-microbe interactions under a microscope. And so I was really excited because I am a microscopist by training, and I was excited to maybe meet other microscopists. But here we are. And so a big part of my project was actually doing FISH, which is fluorescence in situ hybridization. And so I come from a training where seeing is believing. So as much as you can see directly, the better you can believe these phenomena. And so FISH is really powerful because you can start asking questions of who, in terms of bacterium, who is next to each other, who is there, and eventually we want to also decide, image what exactly are they expressing next to each other. And so for this complex application, we turned to classy FISH, which is a combinatorial labeling method, because if you only have 4 fluorophores, usually you're limited to 4 IDs. But then when you start combinatorial labeling things, you get to much more combinations. And so this also starts to exponentially increase as you increase the number of fluorophores. And so one of the first things I did was to do this combinatorial labeling on E. coli just to see whether or not our microscopes can see these different colors. But it also makes for very, very beautiful, stunning pictures. So now that we have the macro and the micro metagenomics, I basically took out the top most abundant bacteria and started targeting them. But one thing that was kind of a thorn in my process was a lot of these genuses, they're actually kind of random. You don't see them. They've only been described in shotgun metagenomic sequencing. And most of you know that this doesn't capture 16S sequences. And so it was kind of a bit like deciding and shooting at the dark. And so one of the first things that we did was actually to do PacBio sequencing using the same shotgun metagenomic samples. And so with this, we were able to get the full-length 16S sequence that is very necessary for my fish design. But it's also pretty exciting because we're able to circularize full genome of previously uncultured bacteria. And so with this, then I align all of these 16S sequences. I use an R package decipher to figure out what parts of the 16S sequences I can target for my fish analysis. And so this is a probe design and just some probes. And what I was really excited about is now you're starting to be able to directly observe bacteria that are in these chicken cica. And so here I'm showing 6 of the top 15 bacteria genuses. And you can now start asking questions of relative abundance and also who is next to each other. One part also, I was really excited about observing this phenomenon where you see these microaggregates of clonal populations. So I had been observing this a year ago and didn't know which bacteria they are. But now I can tell you confidently that Streptococcus and Cacovinus and some other clades also form these clonal microaggregates. And so if you were at Nikolai's poster, his modeling techniques also got to more clumps of bacteria. And so we're excited that different methods are actually converging. And another thing is also these microaggregates are usually brighter. And since fish is a ribosomal targeting thing, it means that they're also metabolically active. And so the idea is that they're forming a metabolite that is supporting their active growth that doesn't really benefit its neighbor. And so hopefully in a couple months or a year, I can tell you a lot more about what metabolites they're making. But for now, these pictures are still incredibly stunning. The other part is, besides doing fish, there's also a lot of heavy image analysis, especially when you start multiplexing. And so for this, I turned to an open source image analysis platform, QPath. And basically, we're segmenting, filtering, and classifying based off of our fluorescence signals, and then we can get quantification. And so one of the good things is when you compare our micrometagenomic data of relative abundance to basically the images that I'm capturing with my research assistant, you see the general trends, which is a good cross-check for both of the methods, because people often ask, like, how do you know your probes are working? How do you know you're targeting the right things? And I know a lot of times people are very nervous when they see very, very large error bars, but in our case, it's actually exciting, because it means that depending on where you're imaging or where you're measuring, there are large fluxes in the relative abundance. And so this means that there is a spatial part of how these bacteria aggregate. So also earlier I showed you some relative abundance correlations, and I just picked out 2 of them where if you map them against each other, it seems like our fish data and image quantification does agree with the micro, which makes sense because we're going to single cell level of imaging. And so here's another example. And also, since we're at a Hologenomics conference, I wanted to end kind of on a bang. And so this is a very preliminary, n equals not much, and we still have to check all of the things. But basically, with our circularized genome and shotgun metagenomic sequencing, we can get to strain level differentiation that fish actually can't. But then also specifically, depending on where you sample, we see this specific bacterium at a strain level. You can see at one kind of more closer to the middle, you see a full, complete genome. But if you sample this same cryosection, but closer to the host, you start seeing these gaps in this same exact bacterium. And we know that they're the same possible strain because their SNPs are all the same, but these gaps are actually prophages and genes. And so what we're really excited to make sure that this is true is that based on where you sample, we start seeing prophage excision or maybe even plasmids jumping out of the genome. And so, like I said, there's so much exciting things coming on the horizon. And with that, I have a lot of people to thank. This project definitely is very ambitious, but it brings together experts from all different types of fields. And so I'm learning a lot, and I also have to thank everyone that's on this list because I couldn't do this on my own. Yeah, thank you.